low fertilizer use efficiency. You must be knowing that fertilizer use efficiency in case of nitrogenous and phosphatic fertilizers. In case of nitrogenous fertilizers, fertilizer use efficiency is up to 50% only. That means if you are applying 100 kg nitrogen, only 50 kg will be available to the plants. And it is even much lesser in case of the phosphatic fertilizers. It is around 20 to 30% only. That means our 70% fertilizer goes waste for the farmers. So which increases the cost of the cultivation also. And why it is happening so? Because our soil microbial diversity has affected very much. Otherwise, there are the microbes which performs the solubilization and mineralization process so that nutrients can be made available to the plants. But our chemical fertilizer application affected this process very negatively. There will be an imbalance between the nutrient mining through high yielding dwarf varieties of the crops and replenishment into the soil. Why this gap is there? Because farmers are applying only NPK. Rarely 5% farmers are using micronutrients. But our plant requires 17 essential elements for its growth. So we are taking from the soil 17 elements and we are giving back hardly 3, 4 or 5. So it will create an imbalance. So it is also increasing the demand on the chemical fertilizers, which creates the pressure at our natural resources like fossil fuel reserves, which are very limited. So we have to think in other directions so that we can manage our crops with the minimum use of synthetic fertilizers. So this can happen with the help of the biofertilizers. So what are the biofertilizers? Biofertilizers are the products containing living cells of the beneficial microorganisms that have an ability to mobilize the nutrients from non-usable to usable form through various biological processes like nitrogen fixation, P solubilization, P mobilization, zinc solubilization, K solubilization, organic matter decomposition, growth hormone production. There are several ways in which they can benefit the plant's growth. So in the market, there are n number of microbial inoculants available for different functions. What are those? Nitrogen fixers. These nitrogen fixers are broadly classified into the 3 categories. One is symbiotic nitrogen fixers. Symbiotic means they need a physical association with the plant. They benefit the plant. They take benefit from the plant. These are highly host specific, like Rhizobium, Azotobacter-evaluation, Frankia, Anabina. Anabina is the cyanobacteria. So they are symbiotic nitrogen fixers. There are many more examples are also there, but these are the most common one. Then among the non-symbiotic category or free living category, Azotobacter, Biorinkia, Clostridium, and there are many cyanobacteria which fix the nitrogen into the free living form also. And one famous example is in the associative symbiotic category. It is neither symbiotic nor non-symbiotic. It is associative symbiotic. It remains attached to the plant roots and perform its function. Azospirillum. Then there are phosphate solubilizers, which makes the phosphate available by several different kind of actions, which we will discuss later. These are the most common examples: Bacillus megaterium, Pseudomonas strata, Aspergillus and Penicillium. These are sometimes asked in the exams also. These are the most common examples for these particular categories. Then there are P mobilizers. Earlier it was P solubilizer, and this is the P mobilizer. We have to keep it in mind that mycorrhiza are potent P mobilizers. They take the available phosphate, which is already present in the soluble form, into the deeper zone of the soil. They take the soil, this available phosphate through the hyphae and transport it to the root of the plant. So the mycorrhiza are P mobilizer. This question is asked several times into the JRF, SRF, and even ARS examinations also, that mycorrhiza act as P solubilizer or P mobilizer. So it is a P mobilizer. Then potassium and zinc solubilizers are also available there. Most common example is Bacillus. Then Burkholderia, Pseudomonas species are also there. Then plant growth promoting bacteria, which produces the auxin, cytokinin, gibberellin, these growth hormones. Fluorescent Pseudomonas, Azotobacter, Azospirillums. Then there are the bacteria and fungi which suppress the soil-borne plant pathogens like Pseudomonas, Bacillus, Trichoderma. These are the most common one. These act as a biocontrol agent for the diseases. Then there are for pest controlling also Bacillus, Metarhizium, Beauveria, Bassiana, Trichoderma. These are the most commonly known examples in their categories. Then compost enhancer and enricher also there. Means if we add these Aspergillus, Trichoderma, we pre-treat our substrate with these fungi, the decomposition process will be very faster. So we will need much lesser time for compost preparation. And compost enricher during the compost curing phase. After the decomposition, after the thermophilic stage, there is a cooling down stage. At that time, we can add Azotobacter, Azospirillum, phosphate solubilizer, phosphate K solubilizer. So which will enhance the quality of our fungus also. Then green manuring like Azolla, Anabina, Rhizobium legumes. So these improves the quality of the soil. Then there are the bacteria. Then there are the bacteria which are useful for the amelioration of the degraded or contaminated soils. Contaminated soils means maybe having xenobiotic compounds, organic compounds, these pesticide residues. So there are Pseudomonas, Xanthomonas, Thiobacillus, hydrocarbon hydrolyzing bacteria like Rhodococcus, Phenolkit, Chrysosporium, white rot fungi. These are used in the amelioration of the degraded soils. Then drought tolerance providing vesicular or vascular mycorrhiza. Mycorrhiza helps in drought tolerance very widely. It forms a thick mantle around the root and prevents the plant root from the desiccation. Then there are microbial pesticides. Common examples are Bacillus thuringiensis. Among the viruses, nuclear polyhedrosis virus. Then Entomophthora fungus, Trichoderma polysporum. Then there is protozoans also, Nosema lactucci. Then microbial weedicides. This is known as Zygogramma bicolorata, which feed upon the Parthenium. It is also known as Parthenium beetle. Then there are the bacteria which can feed upon the xenobiotic compounds and remove them from the soil. Pseudomonas, Clavibacterium, Rhodococcus, Nocardia, Micromonospora, Sporotrichum. These are all the very well-known bacteria. Then coming to some famous one, how they look like on the agar plates, like symbiotic nitrogen fixers, Rhizobium. It is aerobic in nature and gram-negative bacteria. It is highly host-specific, fix the nitrogen only into the leguminous plants. Nitrogen fixation potential varies from 50 to 300 kg nitrogen per hectare per year. Media used for its isolation is Congo red yeast extract mannitol agar. Media is yeast extract mannitol agar, but Congo red is the dye which is added into the media. The purpose of adding this dye is that along with the Rhizobium, there is another bacterium known as Agrobacterium. These two bacteria are from the same family, Rhizobiaceae, and Agrobacterium is common contaminant with the Rhizobium. So colony color of both the bacteria is also same, but what is the difference? Agrobacterium takes the color of the Congo red when we plate our root extract, nodule extract onto the crema plates, and it does not appear. But Rhizobium does not take the color of Congo red, so it appears creamy white or slightly pinkish in the color. So we can observe separately. So it helps in the identification that this is our rhizobial colony. Rhizobium is aerobic bacterium, but the process of nitrogen fixation is anaerobic. That's why they form nodule inside the plant, so they can protect their enzyme, nitrogenase enzyme, from the oxygen and fix the nitrogen very efficiently. Among the Rhizobium also, there are 7 cross inoculation groups, because Rhizobium is very host specific and it infects on the leguminous plants. But among the leguminous plants also, there are the groups like clover groups which infects the genus Trifolium only. Then there is alfalfa group, Rhizobium meliloti. It infects the nodule of the roots of the Medicago mellilotus. Then bean group, Phaseole group infects and nodulates the plant of genus Phaseolus. Then there is lupine group, which nodulates Ceradella. Then P group, Rhizobium leguminosarum, which infect the node and nodulates the pea, sweet pea, lentil, and veg. Then there is soybean group for soybean. Then cowpea group is there for which nodulate the cowpea, pigeon pea, groundnut, etc. Rhizobium of one group cannot nodulate other groups' crops, so they are highly host specific. Coming to the Azotobacter, it is free living and nitrogen fixer. Aerobic in nature, gram-negative bacteria. Its nitrogen fixation potential is 10 to 20 kg nitrogen per hectare per year. The question comes that why Rhizobium is having much higher nitrogen fixation potential, like 50 to 350 kg per hectare, and this Azotobacter is having only 10 to 20 kg nitrogen per hectare per year. Because Rhizobium is free living bacteria, it does not form any structure with the plant. So it have to protect its nitrogenase enzyme from the oxygen also. So it will secrete lot of polysaccharides outside the cell to make the environment anaerobic. You can see the colony size is very large. It is due to the polysaccharide production also.
Hello.
So, hello. Is there any disturbance, sir? Okay. So, the media used for its cultivation is Jensen medium. It is cyst forming, so more resistant to the diverse environmental conditions. This is also very known question. Azotobacter forms this cyst. Also produces the plant growth promoting hormones, and its common species are Azotobacter purpurecum, Azotobacter vinylandi, Azotobacter puspali, Azotobacter salinestris, Azotobacter berenkii. Coming to the azospirillum, which is an associative symbiotic nitrogen fixer, it is microaerophilic in nature. This is also very important. Gram-negative bacteria. It shows the subsurface growth in liquid oblique semi-solid media. You can see here a subsurface pellicle. It does not form pellicle at the top because it is microaerophilic. It requires little oxygen as compared to the aerobic ones. So it grows subsurface. It is not host specific, just like Azotobacter can be used in variety of the crops. Its nitrogen fixation potential is also 10 to 25 kg nitrogen per hectare per year. Because it also does not form any specialized structure with the plants, so it also wastes its energy into the protection from oxygen. Media used for isolation is N-free malate medium. Also, name is of the medium is Omlyansky medium. Then also produces plant growth promoting hormones. Common name Azotobacter lepiferum, Azotobacter brasilensis, Azotobacter palustri, Azospirillum oryzae. So these are the common species. Then coming to the phosphate solubilizing microorganisms. Media used for phosphate solubilizing microorganisms is Picovascia agar, insoluble form of the phosphate. Aluminium phosphates, iron phosphate, tricalcium phosphates, zinc phosphate. These are the insoluble forms which are present into the soil, and these p-solubilizing bacteria acts upon these forms and break the bonds between these metals and phosphates and make the phosphates available to the plants. Mechanisms are like production of weak organic acid, phytases, phosphomonoesterases, phosphodiesterase, exopolysaccharide production, siderophore production, hydrogen ion secretion. See how these mechanisms helps in solubilizing the phosphate. Like we can see here, siderophore production. What does siderophores do? They chelate iron molecule. So phosphate will be freely available into the solution. Coming to the phosphatase, phytases enzymes, they act upon the organic phosphates. They break down the bond between organic compound and phosphate and make phosphates available into the soil. How EPS production helps? EPS chelates calcium ions and free the phosphate ions. Then coming to the acid production, what acid does? Acid also, hydrogen ions also helps in the trapping the calcium molecules. So phosphate will be available to the plant. In the same way, acid forms complex with the this calcium triphosphate, separate the phosphate molecule from the this complex. So in this way, these mechanisms helps in the P solubilization. This is how we can identify that our bacteria or fungi is solubilizing the phosphate or not. So this is the plate containing this Picobus ke agar. It contains the tricalcium phosphate as a insoluble phosphate form. So when we pour the plate, media whole plate is white. After inoculating the bacteria or fungi at the center, if our bacteria or fungi is having the P solubilization activity, it will either produce enzymes or acids or hydrogen ions, whatever mechanism it will use. It will solubilize the phosphate from the surrounding zone and will use the phosphate for its own growth. So, so that we can identify that our particular bacteria is. Sir, am I audible? Hello.
Yes, sir. Yes, sir.
Yeah, okay, okay. Because I did not receive any response, na? So I little bit got worried that whether is in connection is proper or not. Okay.
Yes, sir. You are audible.
So we can identify here that our particular bacteria or fungi is solubilizing the phosphate, because we are not giving any solubilized phosphate in this plate. We are providing tricalcium phosphate, which is insoluble. So if bacteria or fungi has to grow, it needs phosphate. So it will solubilize it first and then it will take for its own use. So this is the nitrogen fixation potential of different system. Legume rhizobium system have the potential of fixing the nitrogen 50 to 300 kg per hectare per annum. Cereals azospirillum system have 10 to 25 kg per hectare per annum. Cereals cyanobacteria system have the potential of 10 to 30 kg per hectare per annum. And rice azolla anabera, which is also very common, it has the potential from 20 to 90 kg nitrogen per hectare per annum. So for these biofertilizers, we need some specific characteristics. Some desirable characteristics are there that so that our desired bacterium can work as a good biofertilizer. So it should be prompt and effective into the nodulation and nitrogen fixation with the host cultivars under the wide range of environmental conditions.
Dear participant, maybe sir had lost their internet connection. He will join soon. Wait for a few minutes.
Dear participants, due to network issue at our resource person's end, he will be joining us within 2 or 3 minutes. Wait for him. Till then, we will have to wait for him. So please bear with us and stay put. He will be joining soon within next 2 to 3 minutes.
Hello, sir.
हाँ ठीक है हेलो सर गुड मॉर्निंग
Good morning, sir.
Good morning, sir. Good morning.
एस्केप कर लेते हैं इसको वॉल्यूम बढ़ा हाँ वॉल्यूम तो है अच्छा बिल्कुल कम है यस 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 बाद हेलो गुड मॉर्निंग सर एम आई ऑडिबल
Yes, sir. You are.
okay so just tell me up to which slide the presentation was continue so that I will start from there only am I ये presentation दिख रहा है ना सभी को is it visible
Yes, sir.
okay so up to which slide we have seen it
Sir, phosphate solubilizing.
okay displayed it was up to here we were continuing hello okay so hello Manohar sir
Yes, sir. बोलिए.
okay so I'm starting from again from this phosphate solubilizing phase sir okay
Okay,
so
okay, fine.
these are the mechanisms which we have discussed that these are the mechanisms responsible for the phosphate solubilization in this we have discussed that cytorophore production helped by chelating the iron molecule so if iron molecules are chelated then it will not form the complex with the phosphate molecules in the same way exopolysaccharide production chelates the calcium ions and acid production helps in chelating the calcium ions then hydrogen ion excretion also helps in chelating the calcium ions phosphatases or phytases break the bond between the organic compounds and phosphorus molecules and make it available to the plants. So when we have to identify bacteria solubilizing the phosphate or not. In that case, we have to pour a plate containing the media.
Sir, the slide is not moving.
Yes.
Slide is not moving.
Yes, sir. It is just first slide we are seeing. Here.
Okay, first slide. Okay, wait. I have to share it again. Then, just a minute. I have to unshare it first. Then, this is the different system. Stop share. So is it visible now? Just a minute. Manohar sir, is it visible now?
Yes, now it is visible. We are seeing the estimate of BNF by different.
Yes. Now you are
Now
able to see
Petri
this, right?
Petri.
Okay.
Thank you.
So these are the mechanisms of phosphate solubilization. Have you heard this slide completely before the network connection?
You may please repeat this slide.
Yes. Okay.
Yes.
Because this is very important slide. So these are the mechanisms which phosphate solubilizing microorganisms takes place. Siderophore production chelates the iron molecules and thereby make it unavailable to form the complexes with the phosphate. So phosphate will remain soluble into the soil. In the same way, EPS production also chelates the calcium molecules and does not allow calcium to form the complex with the phosphates. Acid production also chelates the calcium ions and does not allow to form the complexes with the phosphates. In the same way, hydrogen ion also, it does not allow the complex calcium ion to form the complex with the phosphate molecules. Phosphatases or phytases enzymes, they break the bond present between the organic molecules and phosphorus and make the phosphate available to the plants. So these are the few mechanisms of the P-solubilization. The identification of the P-solubilizing organisms, because we have a bacteria or fungi with us and we have to check its efficiency. So in that case, we have to prepare Picovaschia agar medium. Picovaschia agar contains the insoluble form of the phosphate, tricalcium phosphate. It does not have soluble phosphate molecules. So when we put our bacterium inoculum or fungal inoculum onto the plates, for its own growth, it have to solubilize the tricalcium phosphate. Otherwise, it will not grow because phosphate is very essential for the growth of any organism. So if our culture is phosphate solubilizing, it will solubilize the phosphate of the surrounding zone by any one of the mechanisms which we have discussed, and it will take up the phosphate and it will create a hollow zone around the colony. We can see here the hollow zone, and the diameter of this hollow zone explains the efficiency of our organism. If this hollow zone is of large size, that means our bacterium is very good in solubilizing the phosphate. So in this way, we can measure the efficiency of our bacteria also by measuring its diameter. So coming to the different systems of nitrogen fixation, its potential is varied greatly. Like legume rhizobium system have the potential of fixing the nitrogen 50 to 300 kg nitrogen per hectare per annum. In the same way, cereal azoospermum system fix the nitrogen from 10 to 25 kg per hectare per annum. Cereal cyanobacteria systems have the potential of nitrogen fixation from 10 kg to 30 kg nitrogen per hectare per annum. The same way, rice, azolla, and abina system have the potential of fixing the nitrogen 20 to 90 kg nitrogen per hectare per annum. So our culture should have good characteristics so that it can be a good fertilizer, like prompt and effective in the nodulation. Why prompt? Because there are n number of bacteria which have the capability of nodule formation with the plant, but they are not efficient one in the nitrogen fixation. So our desired bacterium should be prompt in forming the nodulation, and it should have good nitrogen fixation capability and should be able to adapt under wide range of the environmental conditions. It should have good survival on the seed, multiplication with the soil. It should have greater enzyme production to solubilize the nutrients. It should be competitive in nodule formation. So it should not allow others to form the nodules. It should have good growth, fast growth rate, both in media also, in soil also. In soil also, it is necessary to eliminate the competition with the native flora so that it will colonize fast and it will form the nodules. It should have good growth in and survival into the carrier material also. So these are the few desirable characteristics. Then these are the few carriers used for the biofertilizer productions like lignite, peat, vermiculite. Coming to the characteristics of a good carrier material, it should be non-toxic to the biofertilizer. It should have high organic matter content to support the growth of the bacteria. It should be of good moisture holding capacity. It should be easy to process. It should be free of lump forming material so that lumps will not be formed in our final preparation. It should be easy to sterilize by autoclaving or gamma irradiations. It should be available throughout the year in adequate amounts so that the production process does not halt in between. It should be inexpensive. It should have good adhesion to the seeds. Adhesion properties are very important. If it is not having adhesion properties, then seed treatment will not be done with so effectiveness. It should have good pH buffering capacity. If pH changes, our culture survival will be affected. It should be non-toxic to plant also. It should be non-hazardous to environment also. The carrier material can be sterilized by two methods. One is the gamma irradiation and other one is the autoclaving. Gamma irradiation is the most suitable way because the sterilization process makes almost no changes into the physical or chemical properties of the material. The carrier material is packed in the thin-walled polythene bags and then gamma irradiated at 50 kg. This is the unit of gamma irradiation. And by autoclaving also we can sterilize our material. We can pack into the thin-walled polythene bags. Then we can autoclave it at 121-15 lbs psi for 60 minutes. Normally we prepare culture media and we do autoclaving at 121 degrees centigrade, 15 pound pressure per square inch for 15 minutes. But for carrier material it is 60 minutes. So now we have to prepare how to prepare the biofertilizers. Then there are the several steps. First, we have to isolate our culture. Then we have to select our culture, which is the most desirable one. Then culture maintenance. Then we have to prepare the carrier. Then simultaneously we have to multiply our culture. Then after full multiplication, we have to mix with the carrier. Then packaging, labeling, and inoculation. This quality testing, etc. So this is the flow chart which shows how the biofertilizer process takes place. So first we have to take a mother culture. From this mother culture, we have to prepare the inoculum. Then from this inoculum for mass multiplication, we can use either fermenter or large size shakers so that we can multiply our culture. Then we have to, after complete multiplication, we have to check the quality, whether any contamination is there. There should not be any contamination, and the desired cell load is around 10⁸ to 9 cells per ml. So that after mixing, our final product should have around 10 raised to power 8 cells per gram of the carrier. Then we have to mix with the carrier. After mixing, we have to cure it and then pack into the plastic bags. Then we have to store it at 25 degrees centigrade. Then quality checking and storage low temperature finally. So this is the flow chart. These are the different nodules formed by the rhizobium under different crops. We can see here this stem nodules formed by the sesbania. So azorhizobium is responsible for stem nodulation. This is a famous example that stem nodulation is caused by Azotobacter-evaluation calinodans. And these are the nodules found in the white clover, mung bean, chickpea. We can see here the nodules. Then after biofertilizer production, we have to use those biofertilizers into the field. And what is the purpose of applying the biofertilizers? To place a very high population of the inoculant strain into the optimum position for rapid colonization. Seed inoculation, seedling inoculation, and soil inoculation. These three are the methods. So we will discuss these methods into detail. Seed inoculation means seed treatment. We have to take 1 packet of biofertilizer, which is of 200 gram inoculant. We should mix it. For this, we have to prepare a sticker solution first. Sticker solution means 10% of jaggery or guar gum solution we have to make. We have to make a slurry of boiling it. Then we have to cool it. This is the process. First, we have to prepare a sticker solution. We have to boil it to dissolve it properly. Then cool the sticker solution. Then add your culture of biofertilizer to this sticker solution. Mix it thoroughly. Then pour this slurry onto the seed. And then we have to mix it. And after mixing, we have to dry it under the shade. And then we have to sow the seeds immediately. So this is the process. And 1 packet of the inoculant, 200 gram, is sufficient to treat 10 kg of the seeds, or we can say seeds required for 1 acre. Then coming to the seedling root dip, this method is used for application of the biofertilizers into the transplanting of vegetable crops. So the required quantity of azospirillum, phosphate solubilizing or nitrogen fixing bacteria has to be mixed into the 5 to 10 liters of water into a bucket. And then we have to put these seedlings root into this solution for half an hour just before the transplantation. And main field application, in this method, we use 200 ml of biofertilizer per acre. We have to mix it with 400 to 600 kg FIM along with half bag of rock phosphate to improve its quality. Then the mixture of biofertilizer, cow dung, rock phosphate has to be kept under the tree or shade overnight with 50% moisture. And then we have to, this moisture has to be broadcasted or can be applied into the rows during the leveling or just before sowing of the seeds. So coming to the beneficiary crops, Rhizobium is exclusively used into the legumes, but coming to the Azotobacter, we can see that it can be used in cotton, vegetables, mulberry, plantation crops, rice, wheat, barley, ragi, jowar, mustard, safflower, niger. There are the n number of crops which can be benefited by the Azotobacter. In the same way, azospirillum also, it can be used in sugarcane, vegetables, maize, pearl millet, rice, wheat, fodders, oil seeds, fruits and flowers. Blue-green algae is mainly used into the water-loving plants, means fields are comparatively having much more moisture, like rice, banana. Then azolla is specifically used in the rice. Then phosphate solubilizing bacteria and fungi, they don't have any specific host requirement. They can be used in any crops. Then there are the Indian standards. Institution has brought out some standards for different inoculants. There are some Indian standards, then there are international chemical standards are also there. These standards prescribe the quality requirements of the inoculant in terms of the number of viable cells into the carrier material, stipulated expiry period, freedom from the contamination with other microorganisms. The standards prescribe the test procedures also for assessing the quality besides the sampling procedures. Instructions for packing and markings are also given under these standards to ensure the consumer's protection from the substandard quality products. These are the standards, like they should have a minimum population of 10⁷7 CFU per gram of the carrier. They should not have any contamination at 10⁵ dilution. Their pH should be in neutral range, 6.5 to 7.5. They should be having moisture around 30 to 40% in case of carrier-based biofertilizers. Then the particle size of carrier is also very important. This is also a very famous common objective questions. 0.15 to 0.212 mm international ISCU size. So this was about the carrier-based formulations. Coming to the liquid biofertilizers. Liquid biofertilizers are liquid formulations containing the dormant form of the desirable microorganisms and their nutrients, along with the substances that encourage the formation of resting spores or cysts for longer shelf life and tolerance to adverse conditions. The dormant forms, upon reaching to the soil, they germinate and produce a fresh batch of active cells. These cells grow and multiply by using carbon source into the soil or from the root exudates. So these chemical liquid fertilizers have several advantages over the carrier-based biofertilizers, like longer shelf life. The shelf life in case of the carrier-based biofertilizer is only 6 months, but they have 12 to 24 months. There are no chances of contamination as we take the culture directly from the fermenter to the tubes. There is no mixing with the carriers, so chances of contaminations are negligible. There is no loss of properties due to storage up to 45 degrees centigrade. They have greater potential to fight with the native population as they are having resting structures, nutrients along with them, cell stabilizers along with them. Then higher populations can be maintained more than 10⁹ cells per ml up to 12 to 24 months. They can be easily identified by typical fermented smell. Each and every bacterium have its typical fermentation smell. Then there is cost saving, huge cost saving. This is very important on carrier material preparation because it has to be pulverized, neutralized, then sterilized, then packing and transportation. So there is a huge cost involved into the carrier material. So liquid biofertilizers avoids that cost also. The quality control protocols are very easy and quick. We can follow these protocols very easily in case of liquid biofertilizers. Then they have better survival on the seeds and under the soil. No need of running our biofertilizer production units throughout the year because their shelf life is 12 to 24 months. So once we prepare, we can go through 1 to 2 years completely. But in case of carrier-based biofertilizers, the expiry date is 6 months only. So we have to run our biofertilizer unit continuously throughout the year. Then easier in application by the farmers. They can mix with the irrigation water also, they can spray it, they can seed treat directly. So these are quite easier to apply. Then doses are 10 times lesser than the carrier based because the cell load is very high in the liquid biofertilizers. Then they have higher commercial venues as they require less transport charges. Potential is very good. Contamination chances are very less, so quality of product is maintained. So they have very high export potential. Then these are the some well-known advantages of the biofertilizers, like they are eco-friendly. Their use leads to soil enrichment, and the quality of soil improves with the time. They do not show the immediate results. Just like the chemical fertilizers, we apply the urea into the field and next day we can see that our crop is less green. This is not the case with the biofertilizers because it takes the time, but the results shown over the times are spectacular. They harnesses the atmospheric nitrogen and make it directly available to the plants. They increase the availability of the nutrients. They improve the root proliferation due to the release of the growth promoting hormones. Then biofertilizers contains the microorganisms which promote the adequate supply of the nutrients to the host plants and ensure their proper development of the growth and regulation of their physiology. They help in increasing the crop yield by 10 to 25%. They also protect the plant from the soil-borne diseases to a certain extent. This is an example where the yield of bajra was affected by the Azospirillum brasiliensis. We can see here that with zero nitrogen in the same both treatment with or without Azospirillum application, we can see the increase in the yield from 1208 kg per hectare to 1396 kg per hectare. In the same way, we can see with the split dosage of the nitrogen in each and every treatment, the yield was increased with application of the Azospirillum. Then the effect of number of cell load was also studied. If a biofertilizer, Rhizobium biofertilizer was having the cell load of 10 raised to power 7 cells CFU per gram of the carrier, the grain yield of soybean was obtained 1.29 tons per hectare. But when the fertilizer was having the CFU load of 10 raised to power 9, the yield was almost double. So the number of cells are also very important. Then there are the constraints due to which this biofertilizer technology could not get success over the period of the time. There are technological constraints like unavailability of the good quality carrier material, lack of qualified technical personnel into the production units, then infrastructural constraints like lack of essential equipments, power supply, etc. Then the financial constraints like non-availability of the sufficient funds and problems in getting bank loans. Then there are environmental constraints like seasonal demand for the biofertilizers, simultaneous cropping operations, and short span of sowing of the planting in particular locality. Then there are human resource and quality constraints like lack of technically qualified staffs, lack of suitable training on the production techniques, then unawareness of the benefit of the technology due to the problem in adoption of the technology by the farmers. This is the main reason that the farmers are not adopting. Nowadays, the scenario is little bit changing because there are no visual differences immediately shown to the farmer. So they little bit hesitate to adopt this technology. Then there are marketing constraints like non-availability of right inoculant at right time, right place. So this is the most important reason that the biofertilizer technology could not get the success, because we need to develop the location specific strains. Not means that a strain is isolated from the Jammu and Kashmir and it is brought to the Rajasthan, which is quite hot environment, and applied to the field, it will not survive. Or this culture isolated from the temperate zone to the tropical zone. So there are the differences into the efficiency. So if a native or our local formulation is available, that is the better one. This is the first biofertilizer patent which was filed by the US during 1896, named as nitrogen. So there are the subsidy provision also for biofertilizer setup, a subsidy of rupees 40 lakhs or up to 25% of the total cost. दोनों में से जो भी कम होगा, whichever is smaller, it's provided by the NABARD or NCDC. These are the some of the biofertilizer packets we can see here, prepared by the Indian Institute of the Agricultural Research. So this was all about the biofertilizers, what kind of different biofertilizers, what is their production technology, what are their importance, what are their key features. So this was regarding the biofertilizer productions. Am I audible?
Yes.
Hello.
Yes, sir, you are audible.
Okay, sir. Is what okay? There was no interruption in between, sir?
No, sir. No, sir.
Okay, okay. So this was one presentation regarding the biofertilizers. We have a very small presentation on the mycorrhiza also. Shall we continue first? Hello, Manohar sir.
Yes, sir. Yes, sir.
Shall we continue?
Yes, sir. No problem.
Okay, 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 okay. Okay. Just a minute, just a minute. Okay, so slides are visible. Slides are visible, sir?
Yes, sir. Now it.
Yes. Okay, okay.
Mycorrhiza.
Okay. So as I told that mycorrhiza are important phosphate mobilizers. So we will have a broad discussion, little bit detailed discussion about the mycorrhiza. Okay. So slides are moving. Okay, so mycorrhiza is a Greek word which is formed from the mycos meaning fungus and rhiza meaning the roots. The symbiosis is broadly defined as two or more organisms living together. This term was first used by the German researcher A.B. Frank during 1885. This is a very common question. Mycorrhiza is symbiotic mutualistic relationship between the soil fungi and higher plant roots. It is neither fungus nor the root, but rather the structural form of these two partners, which is known as mycorrhiza. So the association of both fungus and plant roots is known as mycorrhiza. Since the association is mutualistic, both the organisms get benefited from each other. The fungus receives carbohydrates and growth factors from the plant, which in turn receives many benefits. Plant receives many benefits from the fungus, like increased nutrient absorption, water uptake, drought tolerance, salinity tolerance, protection from the pathogens. In this association, fungus takes over the role of plants, root hairs, and act as an extension of the root system. Almost 80% of our terrestrial plants are colonized by the mycorrhiza. Plants which do not form the association with the mycorrhiza are Amaranthacea, Brassicaceae, Chenopodiaceae, Crassulaceae. These are the families which do not have any mycorrhizal associations. So this is also very important question. Hyphal mycelium network increases the active root surface area for nutrient acquisition up to 700 times. So you can imagine how much it will affect the nutrient uptake and water uptake efficiency. It will increase so widely. So we can see the benefits where there is no colonization, where there is M colonization. We plant receives several benefits like increased resistance to the foliar pathogens, increased drought tolerance, increased salt tolerance. It also triggers the immune responses the salicylic acid and jasmonic acid dependent pathways. It also increases the nutrient transfer via AMF. It takes the nutrients from the deeper zones and make the nutrients available to the plant roots. It also provides local resistance to the root pathogens, increase the resistance to heavy metal toxicity. So systematic resistance to root pathogens by salicylic acid and jasmonic acid pathways. So in case of the plants which are not colonized by the mycorrhiza, you can see that the nutrients which are present in the deeper zones, there will be a depletion zones in which nutrients are not present, so plants roots will not be able to take up. But the roots colonized with the fungal hyphae, the fungal hyphae will take up the nutrients from that particular zone, and it will bridge this gap, this depletion zone, so that plant can take up the nutrients. Coming to the type of mycorrhiza, so based upon the degree to which hyphae penetrates into the root epidermis, and its further development, mycorrhiza are categorized into the two categories. One is endomycorrhiza and another is ectomycorrhiza. Ectomycorrhiza forms arbuscules, vesicles, spores inside the root cells that further categorize into the arbuscular mycorrhiza, orchidaceous mycorrhiza, ericoid mycorrhiza, whereas the ectomycorrhiza forms the Hurtignate or thick mantle. These are the very important questions for JRF, SRF, ARS point of view. There are some mycorrhizal fungi which form both type of mycorrhizal structures called ectoendomycorrhiza, and which are found in the members of subfamily Ericaceae and Arbutoideae. We can see here the visual differences between ectomycorrhiza and vesicular arbuscular mycorrhiza. In vesicular arbuscular mycorrhiza, fungal hyphae are penetrating the roots cortical cells and making the vesicles, arbuscules inside the plant roots. But in case of the ectomycorrhiza, it is restricting to the outer portion only, making the Hurtignate and a fungal mantle around the root cells. So what are the differences between the ectomycorrhiza and endomycorrhiza? Endomycorrhizas are obligate biotrophs, ectomycorrhiza facultative. Endomycorrhiza penetrates the root cells, whereas ectomycorrhiza does not penetrate. This endomycorrhiza produces its typical structures like arbuscules, vesicles inside the root system, but ectomycorrhizal fungi remains outside the root structure. Endomycorrhiza commonly associated with the agricultural and horticulture crops and tropical trees, whereas ectomycorrhiza commonly associated with the temperate forest trees roots. Endomycorrhiza have a loose network of hyphae in the soil and an extensive growth into the root cortical cells, whereas the ectomycorrhiza form a complete mantle or sheath over the surface of the roots, and hyphae grows out into the cell. Endomycorrhiza cannot be cultured on the artificial media as they are obligate biotrophs, but ectomycorrhiza can be cultivated on artificial media. Endomycorrhiza does not cause morphological changes into the roots, whereas ectomycorrhiza causes the morphological changes into the roots. So these are the differences between ecto and endomycorrhiza. This is also a good subjective question for ARS point of view. Then coming to the arbuscular mycorrhizas, mycorrhizal association produced by the Blue-Meromycota fungi are known as arbuscular mycorrhiza or vesicular arbuscular mycorrhiza, formerly known as endomycorrhiza or endotrophic mycorrhiza. Arbuscular mycorrhiza are the association where Glomeromycetes fungi produces arbuscules, hyphae, vesicles within the root cortex cells. And these associations are defined by the presence of arbuscules. Fungi in the roots spread by linear hyphae or coiled hyphae. Arbuscules are intricately branched hyphae that formed within a root cortex cells. They were named so because they look like trees. Arbuscules are formed by repeated dichotomous branching and reductions into the hyphal width. The vesicles develop to accumulate storage products, and in many web association, they are storage structures for the WAM associations. Vesicles are hyphal swellings in the root cortex that contains lipid and cytoplasm. These may be inter or intracellular. Vesicles can develop thick walls in older roots and may function as propagules. So these are AM fungi morphological features, arbuscules, hyphae, and spores, which we have discussed. So these are the root scans of AM fungi of different rice varieties from my experiment. So I am showing here. We can see here the vesicles and arbuscules here. So arbuscular mycorrhiza. This is the classification. These are two families, Gigasporini, Glomini, and further they are categorized into the Spiculaspora, Gigaspora. Then Glomini is under Anthropospora, Caulospora, Glomus, and Sclerocystis. So these are the classification based upon their spore formulations. Scutellespora, their inner membrane walls are and germination shields. But in case of Gigaspora, these structures, inner membrane walls and germination shields, are absent. So these are the actomycorrhizas. These are the associations between the roots of the around 10% of the plant families. Most woody plants includes the birds. So actomycorrhiza mainly occurs into the woody plants. These are the various structures related with their mycelium, their mantle, arbuscules, articulate root cortex. So what are the functions of mycorrhiza for the plant? So in the soil, they enhance the nutrient use efficiency, nitrogen and phosphorus. They can be used as agricultural amendments, biofertilizers or biostabilizers for the soils. They are also known as environmental indicators. Nutrient acquisition help the plant in resisting against the abiotic and biotic stresses. So they help in the overall development of the plant. These are the detailed things may not be required. So these are the visual differences in that inoculated and non-inoculated plants. These are my experiment pictures, which you are seeing that AMF inoculation have resulted in the growth promotion. You can see here that rice varieties inoculated with the AMF, arbuscular mycorrhiza fungi. Here you can see Satyabhama with arbuscular mycorrhiza fungi. Every rice variety is having better performance as compared to the control. Means with or without AMF, you can see the differences that how arbuscular mycorrhizal fungi helps into the promoting the growth. These are the same things. These are in some details, different transporters which are not required for this presentation. So one more aspect related to the mycorrhiza is glomalin production and role in soil aggregation. Glomalin is the protein secreted by the mycorrhiza, specifically AM mycorrhiza. It is a thermostable protein, so it is an indicator of carbon and nitrogen sequestration also into the soil, and it improves the soil aggregation and ultimately all the soil physical parameters. So these are the main domains of the AM fungi, where we can use the AM fungi in gardening and landscape, horticulture, crops, agriculture, forestry. In each and every agriculture related aspect, we can use AM fungi. So these are the benefits provided under the biotic and abiotic stress. So we can see here different mechanisms performed by the arbuscular mycorrhiza in salinity stress, like increased potassium uptake, decreased sodium uptake, then improved photosynthetic efficiency, accumulation of osmolytes, improved plant water relationships, maintain ionic homeostasis in the plant. Then under the chilling stress also improved photosynthetic efficiency, accumulation of the osmoprotectants, improved plant water relations, improved leaf water potentials, In case of nutrient deficiency, it improves the uptake of the nutrients by intraradical mycelium. Then in case of the drought tolerance, drought stress also, it improves the osmotic regulation, stimulation of the antioxidant enzymes, signal transduction, increase the activity of glomalin to aggregate the soil so that moisture loss will not be much higher, improve plant water relations. So these are the different mechanisms, like for heavy metal stress, absorption to the cell wall, chelation of the heavy metals, their sequestration, then change in the rhizosphere pH and microflora, then immobilization of these heavy metals into the fungal structures. So in this way, mycorrhiza can benefit the plant in several ways. These are some of the mycorrhizal formulations available into the market. So this is all about the mycorrhiza. Okay, sir, thank you. So now the presentation is open for discussion. Everyone is mute. Hello.
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 Yes. So
Okay, sir.
yes. Okay. Thank you, dear participants. If you have any queries, please ask to sir, so that sir will help you out there. Sir, there is one question in chat box.
Okay, just a
Hello.
minute, sir. Just a minute. Just a
Yes,
minute, sir. Yes, I'm. Yes, yes.
Yes, it has been directly messaged to me that
Okay,
so that
okay,
it may not be
Sir,
visible.
you help please explain the advantages of carrier-based biofertilizers against the liquid biofertilizers. Actually, liquid biofertilizers are prepared by observing the disadvantages of the carrier-based biofertilizers. The carrier-based biofertilizers. So if I superior or carrier-based biofertilizers are having more advantage as compared to the liquid biofertilizers, it will be a futile statement, because like liquid biofertilizers are carrier-based biofertilizers are having chances of contamination, lower shelf life, lower cell load, a high cost is also incurred. So looking at all these aspects, these liquid biofertilizers were prepared, in which they have increased the shelf life, they have removed the carrier so that they can increase the cell load also, they can minimize the cost also. So I don't find any advantage specifically associated with the carrier-based biofertilizers. The advantage lies with the liquid biofertilizers. Is it clear? Okay.
Dear participants, any questions if you have?
I think they don't have any questions, sir. Whether they are listening or not, I don't know.
Okay, sir, then we will conclude. So,
But sir, this time very less participants are there. What happened, I don't know.
yes, 13 participants are there. Actually,
Yes,
our
sir.
midterm examinations are also
Yes,
going
yes,
on.
sir. So no problem, sir. You can call me for this
yes.
presentation at some other time also. I will give this presentation, no problem. But during 1st to 10 March, I am having a training at Bhopal. So after 10th of March, I can give, no problem.
Okay, sir.
I will fine tune my presentation also. I will add some more information. So there is no issue regarding that. Okay, sir. Thank you.
Okay, sir. This program is just for up to 10th of the March.
Okay, okay.
And
10th of March.
yes, and our strength is low. basically for microbiology, very few students are admitted every
Yes,
year.
yes, yes,
3
yes,
to
yes,
4
yes,
students
yes.
are admitted.
Yes, that is the probable reason. Yes, because
Yes,
it is covered under the life sciences. Yes, sir.
yes.
Yes, yes. Yeah, it is a
So,
subject specific requirement.
yes. So, on behalf of Postgraduate Institute, Mahatma Phule Krishi Rauri, I am very much thankful to you, Jairam sir, for giving your valuable time and guiding our students on biofertilizer production and different aspects of thereof. Thank you once again.
Thank you,
So,
sir.
dear participants,
I am also very much
yes.
thankful to MPKV Ramuri, as I spent my 4 years of golden period at MPKV Ramuri. That was my base in the agriculture, and I really enjoyed those 4 years like anything. It was the best time which I spent into the agriculture college Dhule. Okay, sir. Thank you. It was very nice, but it could have been better if I would be able to interact with few of the JRF prepares students who are willing to prepare for the JRF. But okay, sir. Thank you so much.